My name is Lucy Chen. This year I did a project about cardiomyocyte maturation in the Van Berlo lab. Cardiomyocytes are the contractile cells of the heart. Adult cardiomyocytes typically have very well-organized sarcomeres, which are the most basic contractile unit of the cell, and extensive and well-organized T-tubules, which are basically in pockets of the cell membrane that allow the whole cell to contract in synchrony. While these characteristics are very important for an adult cardiomyocyte to function properly, many of these characteristics are lacking in cardiomyocytes that are derived from stem cells. This limits their usefulness as treatments for disease and as models for disease. We hope that by studying cardiomyocyte maturation, we'll be able to discover how to fully mature stem cell-derived cardiomyocytes. By staining cardiomyocytes from mice of various ages, we discovered that the T-tubules are almost completely absent 6 days after birth. However, the sarcomeres were already present and relatively well organized. Over the next 2 weeks, the sarcomeres mature completely, and by 18 days after birth, they had completely reached adult length and levels of organization. However, the T-tubules were still relatively immature, although they had grown steadily more organized over the past few weeks.
My name is Stephanie Dung, and I'm working in Dr. Prince's lab this summer. And I'm investigating two proteins found in heart muscle cells called junctophyllin 2 and mitofuscin 2. And they've been previously shown to correlate with problems of the right ventricle heart chamber not functioning properly. And they've also been shown to directly interact with each other. So to investigate this interaction, I've created DNA plasmids encoding for shortened lengths of both of these proteins. And then I've transformed the DNA into E. coli bacteria cells in order to clone and then express these proteins. And the next step will be to purify these proteins in order to eventually map the regions where they interact with each other. We anticipate that gaining better understanding of this interaction between junctophyllin 2 and mitofuscin 2 will help us to develop better treatment for pulmonary arterial hypertension.
My name is Gayatri Dileepan, and I'll be a sophomore this fall at the U. I work in Dr. Ouchi's lab, where we study the relationship between mitochondria and heart failure. As y'all have probably heard of before, the mitochondria are the powerhouses of the cell, the ATP generators. To make ATP, they need a small amount of calcium, but if they have too much calcium, this disrupts the proton gradient needed to make ATP because of their positive charge. And when the mitochondria try to get rid of that calcium by opening up holes in their membrane, they end up letting out some molecules that trigger the cell to undergo apoptosis, or cell death. My goal is to prevent the mitochondria from taking up too much calcium. There's an enzyme which enters the mitochondria and opens up calcium channels. No one really knows how that enzyme gets into the mitochondria, but if we were able to prevent that, then the mitochondria wouldn't be able to take up too much calcium. We've identified a shuttle protein that could be responsible for taking that enzyme in and confirmed that they do form a complex, and when that enzyme's pathway is stimulated, their binding increases.
Hello, my name is Laura Ettinger, and I'm a rising senior at Boston College studying chemistry and biology. For the past 9 weeks, I've been working as an undergraduate researcher in the Dr. Daniel Gary lab. It is well established in the literature that adult mammalian cardiac tissue shows a limited regenerative potential, whereas neonatal cardiac tissue can regenerate fully after injury for a short time immediately following birth. My project for the summer has focused on two unique genes, FOXK1 and MYC-N, that are hypothesized to play a role in cardiogenesis, or the development of the heart in the embryo, and cardiac tissue regeneration. My work for the summer has included in vivo studies with live mice, and in vitro studies including staining of harvested tissue with fluorescent molecules, tissue culture with embryonic stem cells, and a technique called quantitative real-time PCR to measure gene expression. I've been testing 3 working hypotheses this summer. 1, MYC-N promotes cardiomyocyte growth to encourage cardiac tissue regeneration in adult tissues. 2, FOXK1 promotes cardiogenesis. And 3, FOXK1 is a direct downstream effector of MYC-N.
Hi, my name is Elizabeth Enner, and I'm going to be a senior this coming fall at the University of Minnesota. This summer, I've been working in the Kaiba lab, studying the effects of various proteins from the DUX, or double homeobox, protein family on muscle cell growth. The most notable DUX protein is called DUX4, and it causes a type of muscular dystrophy called facioscapulohumeral muscular dystrophy, or FSHD. DUX4 is interesting because it has an activation domain that causes it to kill cells or be cytotoxic, but this activation domain is lacking in other DUX family proteins, like DUXA and DUXB. DUX4 also interferes with muscle cell differentiation, which involves the fusing of multiple cells into a long fiber. I found that adding an activation domain to other DUX family proteins, like DUXA and B, causes them to behave more similarly to DUX4. These results show that the DUX4 activation domain is vital to its mechanism of action and also helps us learn more about DUX family proteins.
Hi everyone, I'm Cody Ho. I'm currently doing research in the Dudley lab over this summer. Approximately half of the 6 million patients with heart failure in the United States have diastolic heart failure, and this is characterized by the reduced ability of heart muscle to relax after each heartbeat. Currently, there's no treatment available for this condition. Overproduction of harmful molecules known as reactive oxygen species, or ROS, have been linked to diastolic heart failure. And these ROS molecules are produced by the mitochondria, the powerhouses of the heart muscle cells. The compound mitotempo targets mitochondria and reduces ROS levels. We modified the chemical structure of mitotempo and tested these compounds to see if they could better reduce ROS levels in the heart. We've identified 2 new drugs that better reduced ROS levels in the heart, and these have the potential to be used clinically as the first drugs to treat diastolic heart failure.
Hello, my name is Jack Ogle, and this summer I worked in Dr. Mary Gary's lab, and in the fall I'll be attending the University of Chicago as a first-year student. My project was focused on the exercise pressor reflex. During exercise, our muscles contract, and our body responds to this contraction by increasing heart rate and blood pressure. These responses are known as the exercise pressor reflex. Previous studies done by our lab and other labs have found that in patients and animals with heart failure, they actually experience an exaggerated EPR. Understanding this exaggerated exercise pressor reflex is extremely important in developing treatments and therapies for patients with heart failure. We chose to study the receptors whose populations are localized on sensory neurons, which are innervated in the skeletal muscle and which mediate the EPR. Specifically, we used immunohistochemistry and quantitative PCR to understand the relationship between TRPV1 and piezo2 receptors, because we hypothesized that these 2 receptors regulate one another in the setting of heart failure. And what we found was piezo2 is upregulated when TRPV1 is downregulated.
Hi, I'm Matthew Q, and I'm going to be a freshman at Harvard University next fall. I spent the summer in the Paralingero lab. My project specifically focused on stem cells that differentiate into muscle cells of the heart. These cells, known as cardiomyocytes, are similar to those found in an adult human heart, but they still have yet to develop completely, and therefore they're not as suitable for use in clinical studies or other treatment possibilities. Therefore, my project looked at several ways and methods to mature these cardiomyocytes for use in clinical studies or other scientific research. By analyzing data from publicly available sets and databases, we were able to hypothesize that certain proteins, when deactivated, would lead to an increase in maturation of these cardiomyocytes. These proteins are known to be regulators in cellular growth and division, and therefore we reasoned that by deactivating these proteins, the cells would not spend energy dividing into more stem cells, but rather they would spend energy towards differentiating and maturing into heart cells. After 2 weeks, we found that 2 of our 5 treatments suggested an increase in maturity when compared to the untreated cells, and we are currently planning experiments on 4, 6, and 8-week-old cells to determine the long-term effects of our treatments.
Hello, my name is Rohan Riker, and I'm going into my junior year at the University of Minnesota. This summer, I worked in the Townsend lab, where we are studying Duchenne muscular dystrophy. Duchenne is a genetic disorder that is characterized by muscle degeneration in the cardiac and skeletal muscle, resulting from a missing protein called dystrophin. During this project, we focused on how an impaired respiratory function can lead to an increase in cardiac injury. We designed an oxygen controller and chamber to modify the oxygen percentage of the air to 10% or less than half that of the normal air. We placed mice in the chamber for 3 weeks, and we are currently measuring the amount of fibrosis in the hearts. We expect to see a greater amount of fibrosis in the dystrophic models that were exposed to a lack of oxygen, as there is previous research showing an increase in cardiac injury to these dystrophic mice. Understanding the relationship between a lack of oxygen and an increase in cardiac fibrosis could help us to develop better treatments to slow the progression of heart disease in patients.
Hello, my name is Matthew Elter. This year I will be a senior here at the University of Minnesota. Over the summer, I worked with the Visible Heart Lab to develop a functional model for cardiac catheterization procedures. So as some background, when a patient comes to the hospital with heart complications, physicians can often use a long, thin device called a catheter to gain access to the heart and perform a variety of procedures. To gain access, they can either come through the femoral artery in the leg, or they can come through the radial artery in the arm, which my project focuses on. So I utilize human cadaver CT scans to model the relevant vasculature from the radial artery in the arm all the way up to the aorta, and then I made a 3D print. So within the Visible Heart Lab, physicians can come in to perform stenting procedures, which utilize catheters on reanimated hearts. And my model can connect to the visible heart apparatus, which is used to reanimate the hearts. And physicians can use this model, which they can use as a training tool, and it will make their procedures more similar to a clinical experience.